Hello, everyone, and welcome to today's webinar, Identification of Frequent Autoreactive B-Cell Response in Pancreatic Ductal Adenocarcinoma Using Cell Sorting and Single-Cell Sequencing. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sony Biotechnology. To learn more, visit www.sonybiotechnology.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. Before we begin our presentation, we have a couple of poll questions that are interactive. You will see your first poll question here in the middle of your screen. Please take a moment to record your response. This first poll question asks, which genomic applications are you actively working on or are engaged with? So go ahead and select the best response for that and click Submit on the screen. I'll go ahead and leave this up for just another moment. If you want to take a moment to respond to the poll question, we greatly appreciate it. All right, we have a secondary poll question here that I'm going to display now. In your current workflow, how do you currently utilize cell sorters? So if you could take a moment to answer this poll question as well, we do greatly appreciate it. All right, fantastic. We have one final poll question before we dig into the presentation. This says, what are the cell sorters where are the cell sorters you use located? If you can go ahead and respond to that one, and then we will jump in. All right. We will have one additional poll question following the presentation. We appreciate in advance your participation on it. But at this time, I would like to now welcome our speakers, Min Yao, Senior Research Scientist, Sanders Tri-Institutional Therapeutics Discovery Institute, and Patrick Murphy, Senior Science and Technology Advisor with 10x Genomics. To kick off today's presentation, Sony Biotechnology will give a quick introduction.
Welcome to today's webinar. I'm Sarah Tafoya, and I'm a Global Product Manager at Sony Biotechnology. Before we dive into our main presentation, I'd like to take a moment to highlight the benefits of using a cell sorter during sample preparation to obtain a higher signal-to-noise ratio in your single-cell analysis. Let us consider two similar workflows for single-cell analysis. In the top scenario, the user generates a single-cell or single nuclei suspension from homogenized tissue. This suspension is then directly utilized for barcoding and library construction, followed by RNA sequencing, data analysis, and visualization. In a scenario without cell sorting, less than 10% of the sequencing reads are associated with intact nuclei and cells. Contrasting this, when researchers introduce an additional step of using a cell sorter to isolate populations of interest, over 75% of the sequencing reads are associated with intact cells and nuclei. Now that you know the advantages of cell sorting, how do you pick the right cell sorter? Let me recommend the SH800 cell sorter. Sony has developed a scientific instrument with an intuitive design and the highest level of automation. This not only enables rapid learning, but also allows for independent operation, eliminating the need for specialized supervision. At the heart of the Sony SH800 cell sorter lies a microfluidic device available in various sizes to accommodate a wide range of cell types, which has been demonstrated to be compatible with various downstream applications. The high throughput of the SH800 enables efficiency and the isolation of rare populations, while precise cell deposition facilitates single-cell, plate-based analysis. All these attributes make the SH800 cell sorter particularly advantageous, especially in handling clinical samples. Clinical samples possess a unique urgency as they arrive unexpectedly and in limited quantities as they become available from patients. Rapid processing upon arrival is necessary to prevent sample deterioration, a task often undertaken by specialists in clinical and molecular biology rather than flow cytometry. Whether it's to process samples right away, to get new users up to speed in 30 to 45 minutes, or to automate your experiments with a true walk-away system, you can rely on the SH800 cell sorter to empower your research. If you have any questions about this incredible tool, you can contact me at sarah.tafoya@sony.com. And now I'm going to give the stage to Dr. Patrick Murphy from 10x Genomics.
All right, thank you, Sarah, for the introduction. As Sarah mentioned, my name is Patrick Murphy. I'm a Science and Technology Advisor for 10x Genomics. And before our feature presentation today with Dr. Yao, I'm going to talk for a few minutes about what's new in the world of single cell from 10x. To make progress in unraveling the biological complexity behind these statistics, 10x Genomics has built 3 industry-leading platforms that each allow you to look at the biology with a unique perspective. On the left, we see Chromium single cell, which is our technology to profile and provide deep insights on dissociated samples. On the right, you see Visium spatial and Xenium in situ. Visium spatial provides whole transcriptomic information with single cell scale while retaining spatial context. You can actually see where the gene expression is occurring within the tissue. Xenium in situ also provides a view directly in the tissue, but provides subcellular resolution on hundreds or thousands of transcripts directly in the tissue section, giving us the highest resolution. And depending on your question of interest in the prior knowledge that you have of your system, one or multiple of these systems may best suit your needs. So I won't really have time to talk about all 3 platforms today, but each platform sort of offers its own unique advantage and particular approach to allow you to resolve complex biology. But for today, I'm going to talk just primarily about the Chromium single cell technology and our newest advancement in this technology, GEM-X. So GEM-X is the next generation of single cell solutions from 10x Genomics. The GEM-X platform incorporates the latest technological advances and is engineered for superior performance. It's completely re-engineered microfluidics and optimized reagents, and it's really going to be the foundation to push the boundaries of performance and new capabilities from our single cell solutions. If you've used single cell from 10x in the past, what you'll see here is going to be a number of significantly enhanced product features and capabilities. So this new version of our single cell technology powered by GEM-X is going to offer substantially increased sensitivity, so you can expect up to 2 times as many genes from your runs compared to the next GEM technology. The assay also has much higher throughput, so now we can have twice the level of throughput per lane, and this also translates then into significantly reduced costs. So we're looking at greater than 2% twofold reductions, in the cost per cell when run on the GEM-X technology. We've made other improvements through the microfluidics that allows us to cut the multiplet rate in half, improving data quality, and we've also been able to increase the cell recovery. So now up to 80% of the cells loaded can be recovered in a run, which is excellent for really low recovery samples, especially some of those really critical clinical samples. And all of this really has been, in part, sort of driven through the redesigned microfluidics to improve robustness and assay performance. Here I'm showing an example of the increased sensitivity that we see with the GEM-X technology. Here we've got two different plots. On the left-hand side showing our 3' gene expression, on the right-hand side showing our 5' gene expression assays. And we're comparing the GEM-X in dark blue to the next GEM, or our previous generation technology, in light blue. And the way that we read these plots, if we look at the x-axis, as we go from left to right, we're getting increased levels of read depth. And as we go from top to bottom on the y-axis, we're getting more transcripts detected. So we can see in the GEM-X technology compared to the next GEM technology, the previous generation, as we do compare these head to head and we go with greater levels of sequencing depth, we're getting more and more transcripts detected with the GEM-X technology relative to the next GEM. In some cases, we're seeing as much as 90% more transcripts detected when utilizing the new technology GEM-X compared to our previous generation. And this ultimately helps us drive greater insights. In addition, we're able to get more insights from our samples through the greater throughput that we see from this particular change in technology. So on the left-hand side, we can see a stacked bar graph showing the different cell populations that are present in a renal cell carcinoma sample, comparing our previous generation technology, 5' V2 next GEM, with our next generation technology, GEM-X V3. We can see the relative abundance of cell populations that are there are very similar, but what we see are significantly more cells in this V3 GEM-X technology. And if we zoom in specifically on the cancer cells, the renal cell carcinoma cells, we can then also look at the gene expression changes. So what we see when we look here at the right-hand side of the plot is that when we're looking at the legacy or previous generation technology V2 compared to our new technology GEM-X V3 in dark blue, we can see, of course, more cells are being detected. And in addition, the genes that we're demonstrating here, we're seeing higher expression of those genes detected. So that's giving us the ability to see both more information from our sample, because we're seeing more cells, as well as more information about the actual transcripts that are present. The improvements aren't just limited to gene expression. If you're very interested in understanding TCR or BCR information, we have made improvements that also increase the ability to capture full-length TCR and BCR information from your samples. So here we have an example where we're comparing our previous generation technology in light blue with our new technology, GEM-X, in dark blue. Of course, we're seeing more cells because we've got that increase in throughput. But if we look at the bar graphs down in the lower right-hand corner, even the fractions of cells that are having full-length VDJ detected and allowing us to identify the clonotypes that are present, we're seeing an increase in that fraction as well, which means that we're detecting and collecting even more T cells and B cell full-length sequences from your individual sample. So with that, I'll thank you for your time. Just want to remind you, we've built 3 different industry-leading platforms that each allow you to look at biology with a unique perspective. Single cell is what I've talked about today, the dissociated sample options. We also have really nice options for interrogating whole transcriptome and targeted transcriptome with spatial context. So if you have a project in mind, let's chat. And with that, I will turn it over to our featured speaker, Dr. Yao.
Good morning, everyone. Thanks again for the nice introduction, and thanks Sony and the 10x Genomics to provide me this opportunity to present our work here. So my name is Min Yao. I'm currently a senior research scientist in the Sanders Tri-Institutional Therapeutic Discovery Institute in New York. The work I present today is the work I did earlier on during my postdoc with Dr. Ductal Fearon in Coastal Harbor Laboratory. The most of the work has recently been published in GSI Insight. So one of the key questions we try to do during the postdoc study is to understanding what the potentially cancer antigen in pancreatic cancer that driving a B cell response. So as many of you are aware that pancreatic cancer is one of the deadliest cancer in US and also in the world. The statistic here is back in 2022, but it's still pretty similar here in these years. So there's about estimate over 60,000 new case, and the estimated new death is actually pretty close to that new case number. So the actually 5-year survival rate is only around 10%. In the right bar, you can see many other cancers have been shown dramatic reducible mortality due to improvement of therapy, especially immunotherapies. However, for pancreatic cancer, the survival rate was continually low. Even immunotherapy have been proved very promising for many other cancers. Immunotherapy in pancreatic cancer mostly didn't work. The only immunotherapy that worked in pancreatic cancer is targeting a very small population called microsatellite unstable or microsatellite high pancreatic cancer, which only represents about less than 1%. And the majority of pancreatic cancer does not respond. While I was on my postdoc study, there's a few landmarking paper come out suggest that tertiary lymph node structure, which typically consists of actually T and B cell response, is correlated a better patient survival in different cancer types, including melanoma, sarcoma, or even breast cancer. And similarly, some earlier study in pancreatic cancer also suggests there also can be present of intratumor tertiary lymph node structures, and that be associated with favorable prognosis factors in pancreatic cancer. And then back in Dr. Fearon's lab, they have been mostly focused on chemokine 6L12 and 6L4 study on T-cell infiltration in pancreatic cancer. And in their phase 1 clinical trial, they interestingly found that when we actively provide administration the 6L4 inhibitor to the pancreatic cancer and also colorectal cancer as well, and then we do biopsy before treatment and after treatment, and treatment only takes about 1 week. And by RNA-seq of this data, surprisingly, we found the 6L4 inhibition lead to a dramatic upregulation of T-cell activation as well as B-cell activation as well. Given the short time window, about only 1 week of this therapy treatment, so we are wondering that there must be a pre-existing B and T-cell activation in cancer that can be further augmented by this 6L4 inhibition. And particularly for the B-cell response, there also the interesting question would be, what will be potentially the antigen that are in pancreatic cancer that lead to such a response? So most of the antigen are currently unknown, especially for pancreatic cancers. Before I go to the detail of our methodology, I would like to give a little bit background of B-cell activation and differentiation. So normally B-cell were generated from bone marrow as a naive B-cell, and they express the antibody on the cell surface. The antibody is also called the B-cell receptors. So each B-cell receptor containing two genes, or heavy chain and a light chain. And in the naive B-cells, the antibody are normally expressed in the IgM, IgD naive isotypes. And those antibodies typically have a low affinity. When B-cell was activated by antigen, in this case, example by a protein antigen here, B-cell will be binding to antigen and activated. And then in this case, this normally happens in germ center reactions. In this case, the B-cell, when binding to the antigen, they will internalize the antigen and then further present this antigen to a helper T-cell called a T follicle helper CD4 cells. And this the critical of this T follicle helper T-cell will further activate B-cell and lead to a germ center reaction. In this case, B-cell were expressing this antibody will be expanded, and also they will have an isotype switch from initially IgM, IgD to IgG, IgA, and IgE, etc. And also there's a somatic mutation in the VDJ region lead to increased affinity of antibody. So there's three features on this germ center reaction on B-cells, including clonal expansion, somatic mutation, and isotype switch, provide a traceable molecular marker on identified activated B-cells. And after a certain high affinity antibody are achieved, B-cell can further differentiate into plasma cell, which is can circulation antibody and the circulation in the tissue and the blood. And some B-cell will further differentiate the memory B-cell and to lead to long-term antigen encounter for same antigens. So taking advantage of those B-cell activation feature, we propose to a pipeline that can lead us to discover a pancreatic antigen that driving B-cell response by first sorting out the B-cell and the plasma cell from pancreatic cancer, and then sequencing the paired B-cell receptors or antibodies. and then screen those antibodies to identify antibodies that can bind into pancreatic cancer, and then try to either produce recombinant antibodies and use this antibody to identify the antigens. We think this approach can provide a multiple beneficial in terms of identify a cancer antigen. First of all, it's a function screening because based on this key feature of a B-cell activation, we can select the antibody goes through clonal expansion, somatic hypermutation, and isotype switch. So those antibody already indicated those are from an activated B-cell response. And this is also an unbiased approach of antigen discovery. So we don't need to per sequencing the genome, and then and also the antigen does not have to be a neo antigen which have a mutation. You can potentially identify abnormal post-translation modification or even self antigens. And because the typical B-cell response require also B and T cell activation, so that antigen we identify not only can induce the B-cell response, those antigen can also induce CD4 T cell response as well. Because we're working with the human samples, so those identified human antibody and antigen can have directly translational applications. So before we started, we decided to first validate this pipeline using an easy mouse immunization model first. In this case, we immunized a B6 mouse with a model imaging of albumin, chicken, we see the chicken protein can easily induce the immune response. In this case, after 2 boost immunization, you can see in the blood, you can see mouse blood, you can see a very nice anti-OVA IgG response. And then we further sorted out those OVA-specific B cells using conjugate OVA, fluorophore conjugate OVA protein. So you can see here that typically present a very small population, like 1.1% of the total B cells. And then the time, this is back in 2017, when Tenex just initiated their chromium 5' gene expression on the human BCR, but they don't have a mouse BCR yet. So we working on our co-facility, John Perez group, we developed a mouse BCR sequencing in combination with the 10x 5' barcode methodology. So we decided a panel of primer targeting the constant region for mouse primer, different isotypes and also heavy chain and light chain. And after we back to the library, we amplify those BCR specifically, and then we sequence for those BCR genes. And then on the right, you see for those who have not seen the 10x VDG sequencing before, so this is a typical VDG browser. You can see, for example, the top clone here, which is a IgG1 clone, have 27 barcodes, meaning that 27 express identical CDR3 region indicates they're likely from the same clone. So you can see we are able to identify quite a few expanded clones. I'll show you the example of top 3. In the lower panel, it may be a little bit hard to see, but you can see that the yellow vertical line, those indicate somatic hypermutation are different from germinalized. So we be able to identify somatic hypermutated and also expanded clones. So we expect 3 of the candidates, IgG, G1, 1, 2, 3 here, and further test this by ELISA to bind into OVA, and then we confirm all the 3 by a very high affinity compared to also another positive control we use and also the negative control. So this provides a first proof of principle. We'll be able to identify an antibody to a specific antigen by this method. We further move to a slightly more complicated models. In this case, we generate a mouse pancreatic cancer liver metastasis model by first engineering a mouse pancreatic cancer cell line MM1 using a Ductal-inducible lentiviral system to induce expression or marker GLP together with the cytosolic OVA. In this case, the OVA only expressed in the inside of the cytosolic region. It's kind of potentially mimic a more natural or occurring antigen situation in cancer in general. And we inject this through a tail vein, portal vein, and that tumor will grow in the livers. So we divided our mouse in 3 different groups. One group without Dux, so all the antigens are not expressed to serve our control. And then one group, we let the tumor grow 1 week and then induce Dux for 2 weeks. And the third group, we let the tumor grow 1 week and then induce Dux for 3 weeks. And then we monitor the antibody response by sampling of blood from the tail veins. And you can see here, for the control, for no Dux or wild-type mice, you really didn't see much antibody response to OVA. And then with the Dux on for 2 weeks, you began to see some low antibody response to OVA titer on IgG, but the dose was on for 3 weeks, we do see a very high titer of overexpressed IgG response. Surprisingly, the GLP, turbo GLP here is also another cytological marker we use. We unexpectedly detected we also have a very strong titer response to antibody response to binding to the turbo GLP. So we further analyze those immune cell population within the tumor and also draining lymph nodes. One slide we're showing here is analyze plasma cell, actually particularly plasma blast cell, marked by CD19 and CD138 double positive by across different compartments, including tumor without dogs, with dogs 2 weeks or 3 weeks, or in liver non-tumor area with normal control, and also in draining lymph node liver or a more distant spleen site. And one thing striking come out here is that we used a spike of plasma blast cell after we dosed after have a dose for 2 weeks in control of no dose control, and somehow with further longer this plasma blast cell were recline of a longer time if we have dose for 3 weeks. And then we want to further confirm whether those plasma cells indeed have antibody secretion reactive to OVA or turbo GLP. In this case, we develop ELISpot assay. In this case, we place the plasma cell on the ELISpot and detect whether they react to secreted IgG antibody by the total IgG blood, or separately we detect whether it can secrete antibody binding to OVA or turbo GLP and quantify the percentage of cells. In this case, we found that, as the example showing here, about 4% of IgG producing plasma cell have antibody response to OVA, and about 11% produce antibody to turbo GLP, which is kind of similar to what we see on the serum titer ELISA. We actually, in this case, we further follow up, had a collaboration with the 10x. At the time, the 10x just released their mouse PCR kit. So we are their beta tester, and we further actually sequencing the oldest mouse model on the B cells and also the plasma cell on the VDJ sequencing. And we actually also sequence of some of the clones to confirm we'll be able to obtain monoclonal antibody binding to OVA or turbo GLP. And those data will not be presented here due to the time limit. So with those pipeline validation, that gives us confidence to decide further move on to studying human pancreatic samples. So we started collaboration with Northwell Hospital, where there are biospecific department there. So we're able to obtain naive treated pancreatic cancer. The reason we especially request naive treatment of pancreatic cancer because mostly pancreatic cancer can be neoadjuvant treated with chemotherapy, and we worry chemotherapy can kill some of the immune cells we are interested in. So we collected 4 resection tumor and 3 fine needle aspiration samples, and the picture on the left show example of a fine needle aspiration. Typically, we will give a small proportion of samples, and for resection tumor, it's hard to obtain, but we do a bit to get 4. And then from there, we started. We process most of the sample to do cell sorting and single cell sequencing, and we also try to obtain some sample to try to generate a paired pancreatic cancer organoids. Or kind of some mentioned earlier, in this case, the Sloan sorter actually can be very helpful for us because although we work on the pancreatic cancer sample, particularly on the resection tumor, normally those surgeries take a very long time, 3 or 4 hours in general, and they can when they come to us and also take another 1 hour on taxi, and then so it's normally when sample comes pretty late, and then after we did all the digestion and the processing, it's already over late in the night, and most time the only sorter we are available to use is the only sorter which we can do unassisted author usage, and also our core single cell core also give me a facility to be able to run this single cell on their Chromium machine by myself. Most of the time, there are special traders only controlled by their own core user. So in this case, I showed an example here. We did analyze all those tumor cells and then sort it out and for the single cell sequencing. For the sequencing, we take advantage of the barcode that we can sequence both the gene expression profile as well as the BCR together, and so they will be linked together. This provides us the opportunity to further understand the better immune microenvironment as well as our interested antibody sequence on the BCRs. So we did a single cell sequence in each of the samples separately. Also, the picture here, we project here is the aggregated sample from the 7 human samples. You can see the color indicated. So we see a dominant population of T cells, and then followed by myeloid cell, And we do see a appreciable proportion of B cell and the plasma cell there. And on the right, you can see the distribution in individual patients. Again, you see some variation, but generally we do definitely see a dominant population on T cell and the myeloid cells. And this population detected by single cell sequencing generally agreed well with what we see earlier on by flow cytometry. So we decided to move forward and sort of phenotypic analysis with those immune cell population. And I will first describe here about the T cell phenotypic analysis. As shown here, T cells were defined by CD3, although CD4 and CD8. So CD4 genes segregated very well here. We can potentially further segregate it by a computation analysis. But when we look at the potential phenotypic marker of CD8 cell, we do see quite many cell express the activated CD8 T cell markers, such as perforin and granzyme B, which are very critical for more CD8 T cell killing. And also including a few key activation cytokines, including interferon gamma and TNFs. And similarly for CD4 T cell, we also see the expression on many other markers, such as in the bar graph in the bottom. I didn't highlight the heat map picture, including the CD4 express a key of a Th1 cytokine profile, including the transcription factor TBX21, also cytokines TNF and interferon gamma. For us, we're also particularly interested to see whether we're able to see some potentially T-activated T follicle helper T cell, which present a marker such as the 605 and the BCL6, and also some CD40 ligand on the CD4 population. And we'll be happy to see some, so the population generally are pretty low. Another thing I forgot to mention here, we do see in this T cell population, we do see presence of immunosuppressive markers such as PD-1, CTLA-4, and also regular CD4 T cell, FOXP3 present CD4 T cell. It's representing about 20%. So in general, the T cell do express shows activation status, but also there shows some potential immunosuppressive feature as well. When we look at B cell and the plasma compartment, for example, here B cell was highlighted by expressing CD19, also CD20 as well. There's some populations still express a naive B cell marker such as IgD, but we can see that many also express ISAP switch such as IgG1 here. In the B cell population, we also see some of the express activation markers such as CD27, CD80 on the bottom, and also CD40s. And particularly for our interest, also nice to see some germ center B cell markers expressing BCL6 and also AID, which is the key enzyme involved the ISAP switch. In the bottom panel, you will see it's a marker on the plasma cell. The plasma cell are featured by high expression of immunoglobulin gene, for example, by Ig-kappa constant region. You can see the heat cap here is much higher compared to what you see in the B cells. And then also some other plasma cell marker, GHM and SCDC1, PRDM1, also most of the plasma cell express very low level of CD19. And when we look at also the cell trafficking markers, so interestingly, we found both B cell and then plasma cell expressing the mucosal homing factor interferon alpha, integrin alpha 4 beta 6. So that's as you can see here. And in addition, the plasma cell also express CCR10, which is also a marker for mucosal homing, but not as intensity homing marker CCR9. So together, those data suggest that we do see activated B cell and the expansion of plasma cell population, and the surface marker indicate those B cell and plasma cell show the mucosal trafficking marker, also kind of try to eliminate the possibility that those may come from blood contaminations. Lastly, we also looked at the paired histology slides from the pathology from our clinical collaboration from the same sample of the pancreatic resection tumor samples. In this case, for the two examples here, you can see the dense lymphocyte aggregate structure on the edge of the tumors. So it's kind of like a tertiary lymph node structure, lymphocyte structures. We actually quantify those from all 4 resection tumor. We'll be able to identify all those tertiary lymph node structures in every patient. So combined with the gene expression profile, those data suggest we do see activate T and B cell response in pancreatic cancer, and suggests to potentially suggest a likely ongoing immune response. And also, I want to point out here that all these 4 resected tumor will also be able to do the MSI status test, and all they are confirmed to MSI stable pancreatic cancer. So they are representing the majority of pancreatic cancer. So we next sequencing the VDJ from the same samples using the paired VDJ sequencing. So most of our sequencing data was coming from the plasma cell. Generally, they have a higher expression of hemoglobin genes. The B cell VDJ will also be able to sequence some. So those B cell sequencing, the sequence of B cell with the paired VDJ are smaller, and generally, so there are most of our focus here is focus on the plasma cells. And you can see here, we generally detected plasmas with a paired heavy chain and light chain from each patient. So one patient, we don't need to take any samples that was included for further studies. And when we look at this, the isotype in each plasma cells, you can see only a small population, about around 5 to 10%, express the original IgM isotype, and the majority is already switched to IgG or IgA, depending on different samples. For example, from patient 8 and 19 or 20, mostly are dominated by IgG subtype, while in patient 14 and 15, they are dominated by IgA subtype, excuse me. When we analyze the somatic hypermutation on the VDG regions, you can see they also on the both heavy chain and light chains, they have showed a very extensive somatic hypermutation with area about 20 in both heavy and light. And this mutation level was comparable to published literature on people who go through like influenza immunization or go through rotavirus infections. So in the case those plasma cell have go through likely T cell dependent B cell activation and then somatic hypermutation selection process. When we look at the clonal type of those plasma cells are showing for the 6 sample. For 4 of the 6 sample, we detected expanded clone types are shown by figure A here. For example, in patient 8, you see that the dominant clone have 20 plasma cell have a paired heavy chain and light chain, and followed by a few smaller clones. In the patient 19 and 20, actually half of the plasma cell are in the expanded clones. And in the right, I'm showing some example when we looked at the individual heavy chain and light chain. Actually, that turned out that the clone type of defining A actually is a little bit underestimated, because in the right we actually see in many cases there are much more cell express the same heavy chain or same light chain, but some of them are not paired. So that's why. So for example, for patient A-3 antibody, we actually have a 21 heavy chain and 21 paired chain, but the clone type are going to give out 20 because there's one additional heavy chain and one additional light chain that are not paired. And so most of the if you look at the lineage analyzed based on somatic hypermutation on the heavy chain or light chain, so most of the clones are clonal. So you can see the big circle here, and a few occasionally we see additional further evolution on the somatic mutation among branches, small branches. So again, together with the earlier VDG analysis on the isotype and the somatic mutation, those give us conclusions that the plasma cell in the pancreatic cancer environment have go through activation feature of B cells, the clonal expansion, linear isotype switch, and also somatic mutation. So we have to further understand what those antibody, those plasma coated, what their reaction are, what the potential antigen driving those response. So we decided to select about 41 antibody from all 6 patients, and we choose to select antibody from each patient, including each patient, and those antibody are synthesized or recombinantly using initially on human IgG1 platform, but later we also switched to IgG4 with the histag to potentially aid us in potential screening on binding to tumor tissues. So those recombinant antibody was synthesized with 293 cells, and we will further screen their binding to cancer cells, cell lines, organoids by different methods, including immunofluorescence staining, Western blot, ELISA, tumor cell ELISA. In our empirics, most of the immunofluorescence staining give a better screening capacity and also sensitivities. So that's most of the results are represented from immunofluorescence staining. So this is an example of our staining of different pattern of antibody staining with 2D cell line MIA-PACA2. This is the staining, the fixed and permanent staining. So the overall impression is interesting. Most of the antibody binding to intracellular antigens. Actually, all the antibody we screen here, the positive binder all bind to intracellular antigens. And they are showing different features of binding. For example, the first antibody 8-3. By the way, the number of the antibody name is a little random, but they have some pattern. The first letter indicates which patient is from. So this antibody coming from patient 8, and the third antibody from this patient. So you can see this antibody actually very interesting cell surface-like structures, and later we confirm this structure actually is inside of the cell, so it's likely on the cell cortex. And this antibody also is the biggest clone in this patient 8, so the clone size of 20 is the IgG2 antibody. And similarly, we have a different antibody, second antibody 15-7 from patient 15 is a single clone antibody. Is recognized in some organic structure in the cell line. And then we have a third antibody from a patient 19. It's also one of the biggest clinic clone in the patient. This clone has 46 plasma cells, and they recognize a very interesting structure in the cytoplasm. And similarly, in this patient 19, we see some other antibody 19-4 or 19-3. They stain different structure. 19-4 equally stain both nuclear and the cytoplasm, where 19-3 mostly stain on nuclear structures. So as we summarize it on the right, so out of 41 antibody, we detected a positive binder of 25 antibody. So it's more than half of the all antibody screen shows the positive binder. I forgot to mention that for our 41 antibody selection, we mostly use the expanded clones, especially the top expanded clone. For the samples which we don't have expanded clone, for example, patient 14 and 15, we also express a single plasma cell, and also as well. So if you look at the on the right, the bar graph A, so you can see if we look at the original antibody, the clone they generally the bigger clones they are show the more high likely that will bind to screening. So the positive binding on the screening, and when there are single clones or smaller clones, the binding event, the positive ratio was lower, but still even in the single clone, we detected about half of the antibody still show the positive binder. And all the 25 antibody are show the binding to intracellular antigens, and you can see they are ranging from cytoplasm, nuclear enriched, or cytoplasm enriched, or equal distribution between cytoplasm and nuclei. So we decided further, since the first data I showed you is just screening result on the one cell line, to see whether this binding was specific to more general to other cell lines as well. We also screened this antibody with different cell lines, as well as a normal pancreatic cancer cell line, and also non-stromal cell line. For example, here, the antibody 8-3 binding to a cell line showed earlier, MIA-PACA2, but also binding to similar structure on SUP2 and PACA1. So all three unrelated pancreatic cancer cell line, and there was also the normal pancreatic cancer epithelial cell line, Ph.D cell line. Interestingly, the antibody also binding fibroblasts, which is presented predominantly in pancreatic cancer stroma as well. Similarly, the second antibody 19-3 is binding to nuclear structure in all the 4 cell line was screened. We also looked at this antibody on staining on the tumor section from the patient we already derived. Remember, 8-3 coming from patient 8, and then we co-stained with the tumor cell marker carotene 19. And interestingly, we see this antibody stained both the cancer cell as well as the stromal cell. Similarly, the antibody 9-3 also stained this nuclear structure in this paired patient 19 tumor sample on both tumor cell and the stroma. So in the paper, we also presented the screening on the other antibody as well that also shows similar strategy. So it seems that those positive hidden antibody, the binding to antigen expressed both in cancer and normal indicates that they are likely or unmutated self-antigens. To gain further insight of those, we take an effort to try to identify the antigen to recognize by those antibodies. And we are successful to identify 3 out of 25 antibodies for their antigen. And I will show you there's 3 examples. So the first antigen, to identify the antigen, we typically use antibody to immunoprecipitate the cell lysis and then run a surface staining. In this case, example shown by antibody 8-3, when they pull down, so here, out of the antibody heavy chain light chain, you will see a band here on around the 42 kilodalton band, and also a high molecular band around the 260. This is by surface staining in compared with isomer control antibody IP. And then further, you can further increase the stringent of IP by increasing the sodium chloride concentration during the IP and the washing buffer. In this case, even when we increase sodium chloride up to 500 millimolar concentration, we still see this detection with this band. Further, by cutting out of this band, we'll be able to identify the 42 kilodalton actually represent actin, and the higher molecular weight equal to actin binding protein called myosin 9 and myosin 10. When we co-staining with this antibody again with a actin F-actin marker fluoridine, you see they shows a nice co-staining, and also shows the co-staining with the myosin marker myosin 10. When we treated the F-actin network with the disabled drug cytochalasin D, in this case the F-actin was destabilized. Now you can see the by fluoridine staining and also our antibody AZ3 staining, they are still co-localized on after with the destabilized F-actin, but then they're no longer correlated with the myosin 10, indicates likely F-actin likely the candidate. We further decide to purchase actin, or actin actually is stated in two different states. The monomer states called globule actin, and the polymer form called filament actin or F-actin here. So we generated in vitro using polymerization or depolymerization protocol to generate the polymerized F-actin or a non-polymerized G-actin format, and then using dot blot assay. And as a positive control of the commercial actin antibody, we recognize all the actins. You can see there, they recognize this either F-actin, G-actin on the titration when we titration the actin, we blot it on the blot. However, our antibody AZ3 more specifically recognize the F-actin than the G-actin, and the isomer control doesn't recognize both. We further develop a F-actin ELISA protocol, and we can measure the binding affinity. In this case, we measure the EC50 about on F-actin around 9 nanomolar range. And then we further confirm this by genetic actin is the essential gene, so we do the siRNA knockdown. When we knock down actin, in this case, you will see a decrease in fluoridine staining, but also decrease in our antibody AZ3 staining. But in comparison, if we knock down myosin 10, our staining on fluoridine and also our antibody AZ3 was not affected. Further confirm antibody AZ3 indeed recognize the F-actins. And similarly, we identify a second antigen for antibody AZ3, which is also a big expanded clone in patient 19. IP pulled down a protein band around 55 kilodalton, and the mass spec identified is a complex containing RUBR2 and RUBR1. And the full name is the RUB-like protein 2, is a DNA helicase, and the RUBR1 or RUBR2 typically are working together in a protein complex. And the co-staining with the RUBR2 shows that indeed co-localized in the cell lines. And when we use the recombinant protein, we found this, our antibody only recognized RUBR2, but not recognized RUBR1. Luckily, RUBR1 is pulled down by interaction with RUBR2. And our antibody also recognized this recombinant protein. We measured this affinity, so the affinity turnout is pretty low, about EC50 around 0.2 macromolar. And then we further confirmed by knockdown RUBR2 and showed the antibody staining with a decrease. And for our third antibody, which I showed it earlier example, this antibody 15-7 recognized a very interesting structure in the cytoplasm, look like some organelle. So we did a panel of screening of different organelle markers and found this antibody co-localized with the mitochondrial marker such as the COX4A1, another marker HSP19, HSP9, though HSP9 also expressed in the nucleus. And then our initial trial with the IP with the antibody didn't work well. But unfortunately, this antibody is one of the rare antibodies that can recognize a band in Western blot. And when we did a fraction of a mitochondrial fraction, we found that this antibody recognized a enriched band around 60 kilodalton. And we cut this band for mass spec, and then we identified potential candidate called HSPD1. And when we first studied with this antibody with HSPD1, they indeed co-localized. And then when this antibody also confirmed to binding this HPD1 by using recombinant protein ELISA, or also knock down these targets. And HPD1, for your information, is a heat shock protein. It's called a 60 kilodalton heat shock protein, especially localized in mitochondria. So I showed the example that we'll be able to identify 3 antibodies to recognize the antigen that all shows the unmutated separate proteins, including both F-actin, RUBR2 nuclear protein, and also mitochondrial protein. Because those antibodies are coming from individual patients, we want to know whether this antibody response can be general in pancreatic cancer as well. So in this case, we collaborate with the 2% lab and obtained a cohort of a serum sample from 59 pancreatic cancer or 61 normal health control donors. And the interesting, we found the antibody response, IgG response to F-actin, is also significantly increase in pancreatic cancer, as well as, and also another antibody response to HPD1 also significantly increased. So the antibody response to RUBR2 was not. So this indicates at least 2 of the antibody antigen response likely is maybe common to pancreatic cancers. At the time when we identified those antigens, we looked back to the literature, and surprisingly, we found actually back in 2012 that the research paper, and actually from later a few other several papers from the same lab, as well, showing that F-actin can be function of the evolutionary conserved damage-associated molecule because they can release after 30 days. And actually, they found that the receptor called DANG1 or silicon-9A recognition of this F-actin can lead to inflammation in cell to kind of alert the tissue that the tissue damage happens. In a separate paper in breast cancer, the paper they also identified in breast cancer there's some B-cell response to the F-actin. And similarly, in autoimmune disease, there have been known that in autoimmune disease, there's a many auto response to self-antigen, including in F-actin. In this case, is the type 1 autoimmune hepatitis. The detection of F-actin autoantibody in pancreatic cancer is the first study that actually reports that. And similarly, there's also reports on autoantibody response to RUBR2 and HPD1 in other cancer type, but I think we are the first identify they are showing the induced B-cell response in pancreatic cancer. So in summary, here we showed we have been developed and validated the pipeline to identify B-cell cancer antigen in pancreatic cancer in the mouse model. And in the human samples, we showed by single-cell sequencing profiling, we showed the accurate B and T cell response and expanded plasma cell clones. And our antibody screening to identify most of the antibody, there's a common response of antibody from plasma cell, they recognize the intracellular antigens. And out of the 25 positive hit, we're able to identify 3 self-antigen, including F-actin, RUBR2, and HPD1. And lastly, we showed some of the antibody response, F-actin and HPD1 is common to other pancreatic cancer as well, and that showed significant increase in the serum titer in the cohort that we analyzed. And we hypothesized that during the pancreatic cancer progression, the intracellular antigen may be released during cell death in combination with the chronic inflammation context that may lead to germ center B-cell and T-cell interaction and lead to B-cell activation and further differentiate the plasma cell have produced such autoreactive antibodies. The one question we have not addressed is whether those autoreactive B-cell responses have a role in the pancreatic progression, and that's something we'll be interested to follow up in future. With that, I would like to thank my mentor, Dr. Fearon, for supporting me for this project, and many people in the Fearon lab, including a few I had here, are actively involved in this project. As you can see, this project is active collaboration with multiple partners, both through the Northwell tissue donation program for the surgeon who prepared the sample, and also quite a lot of community within Customer Harbor, including John Proel, who is running the single cell facility, and Johannes Ye, who helped me produce recombinant antibody, and Dr. Pampain, who runs the mass spec facility, and the Tucson lab helped with the collaboration on the pancreatic cancer serum cohort, and also organic culture. And also our collaboration with the CIPO lab helped me do some of the computation analysis on the lineage tracing analysis. And this was also funded by Simons Foundation and then the Last Garden Foundation. Again, also I give thanks to 10x for their sponsorship for the original the mouse BCR sequencing. With that, I would like to stop and take questions. Thank you very much.
Thank you, Min and Patrick, for your informative presentations. Before we begin the live Q&A portion of this webinar, we have the one remaining poll question that will be displayed on your screen. Please take a moment and respond to the poll. It asks, would you like to learn more about the Sony cell sorter and its capabilities discussed in this webinar? If you can please just submit your response. All right, wonderful. And while we have that up, I'm going to go ahead and ask our first question. It looks like it is for Patrick. Patrick, this question says, you showed the new architecture demonstrates improved TCR detection. Is there a change in BCR detection or sensitivity?
Hey, thank you very much for the question. So yes, I did show, just the TCR side, but we do also see a similar increase in the sensitivity and detection capabilities for the BCR, B-cell receptor side of things. So with the new architecture, we do capture a greater number of unique transcripts for the BCR, which aids in the ability for actually reconstructing the full length heavy chain, light chain, kappa chain. So we do ultimately get better recovery from the BCRs as well with the GEM-X architecture.
Wonderful, thank you. And I think this next question is also for you, Patrick. This question says, if I already have data generated using the previous generation, can I switch to the new generation now, or should I wait?
Yeah, also a very good question. Thank you for asking that. So if you've got previous generation data, so the next GEM technology, you can start moving over to GEM-X if you'd like to. We have a data harmonization and aggregation algorithm in Cell Ranger that allows the 3' V3 to go with the 3' V4 in the next generation. Same with the 5' side of things. We'd probably like to have a more specific discussion about how far along the project is, how much data you have on the existing platform, because a lot of people, we're encouraging to sort of stay on the original technology you've used unless there's a really compelling reason to switch to GEM-X immediately, and then ultimately just start GEM-X, the new technology, new version, with any new projects you have going forward. So that would be a great thing to reach out to me if you have any questions on that, or someone at 10x, we can provide more technical assistance about your specific project.
Wonderful, thank you. All right, our next question that we have here, I believe is for Min. That question says, how do you ensure the viability of cells or the integrity of nuclei when sorting samples with the SH800 cell sorter?
Okay, that's the question. So in this case, we sorted the live cells, so we use the cell level BD dye to include the dead cells.
Okay. Our next question here, this one is also for you, Min. This says, did you find any antigens that were localized to the cell membrane?
Yes, that's our original goal. Unfortunately, we didn't. All our positive hits are recognized intracellular antigens.
Great. All right, our next question here for you, Min, that says, considering the complexity of the tumor microenvironment, how did you ensure the accuracy and reliability of the sequencing results?
So that's a good question. So first thing, we try to only this case only focus the immune cells. So when we sort, we actually get it at the non-immune cells such as epithelial cells, and then we did actually flow. When we did the sorting, we also did a flow, a panel flow. So we kind of correlate our flow result with the sequence result, make sure they are relatable. Actually, in our supplementary data, we do show the sequence data population ratio generally correlates well with the flow data. So that's another way for validation.
Okay, great. Our next question here that we have says, did you observe mutations in the 3 self-antigen genes in the cancer patients from which the antibodies were generated?
That's also another very good question. So we can conclude it's likely that's generated by self antigen generated by like autoimmune response. But for the 3 antigen, we identified the F actin, HCD1, and the RUBR2. So for F actin, that patient, we are fortunate enough to generate paired patient organoids. So we did sequencing on that genes, all the actin genes, and we didn't find any mutation on the coding region. So we're sure for the F actin that it must come from a non-mutated self-antigen induced autoresponse. For the other 2 antigen, we don't have organoid sequencing, so we cannot confirm that. But given that that 3 antigen autoresponse have been detected in autoimmune disease and also some other cancer, I think likely they are self antigen driving.
Wonderful, thank you. We have time for just a couple more questions, so we're going to squeeze them in here. Our next one says, are there any common features among the identified self-antigens that induce an autoantibody response?
This one a little bit hard, I guess. So since we only be able to identify 3 antigens, of course, the first thing I think seems the aura intracellularly. Since we only identify 3 antigens, so it's interesting when we look at those antigens. It occurred to me all the 3 antigens actually are form of polymer in their function. Definitely affecting the polymer, but also HCD1 and the RUBR2 do function in a heptamer or octamer complex. So likely, I think those polymer complex likely can lead to break the immune tolerance.
Wonderful, thank you. And it looks like we have time for just one more. So that question is, have you investigated autoantibody responses to the antigens identified in other investigated autoantibody responses to the antigens identified in other types of cancer?
Yeah, we did a little bit of look at other cancer types, including ovarian cancer and the colorectal cancer. Unfortunately, the 3 antigen we identified with some ELISA with the serum titer with a cohort of similar lab, but we didn't see the seems that somehow in this case they're unique to pancreatic cancer.
Okay, great. And we had one more question that just popped in, so I'm going to ask that really quickly. That question says, have you done any experiments that correlate antigens with response to immunotherapy?
That be our ideal experiment we want to do, but unfortunately we haven't done anything with that. For the only 3 patients, we don't have enough time window to follow up to see how they're going. But as I said, they are all MS stable, so they are not for that 3 the patients. They are not able to go through immunotherapy in clinical application.
Wonderful. Thank you again, Min and Patrick, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.